Hey, we're here. So, this would be part two of our conversations. I did like a three-part, I broke our last one into three parts. And I was disturbed because all the video that I was showing you while I was talking to you, none of it went into Zoom. So I had to I had to go in and edit the whole video. So anyway, hopefully this will go better. So how are you doing? So what do you want to go by? CBrown,
CBrown is
Craig,
cool.
what? Okay,
Yeah.
okay,
hmm
that's good. Okay, good. Call you C Brown, Sub Brown. Uh, anyway, so the conversation is about junk DNA, and I I'm not fond of the. Now, the interesting thing is I've studied this stuff all my life, and I'd never heard of junk DNA, and I never heard of junk DNA until I got into YouTube and started listening to Standing for Truth in like 2017, 2018. And that was the first time I ever heard the term junk DNA. So, and and I had molecular biology of the cell. I've got like 4 or 5 different editions of that book, and I know they mention it in the latest edition a couple of times. But so it was never a big thing that we really thought about. So this dichotomy between junk and functional it's not a dichotomy. It isn't one or the other. It's a gradient between the two, according to my thinking. And lately, I've adapted the term junk drawer DNA because I understand that what I would consider non-functional or neutral DNA occasionally is comes into play and later in the future in a species, and it ends up becoming a part of the functional DNA of that organism. So that's kind of my understanding of it. But I wanted to first of all try to get some parameters here for try to explain to you why I believe that there is highly unfunctional DNA, non-functional DNA in the genome. Okay. And what is your position on this? What do you believe?
Okay, so yeah, that's actually kind of pretty informative because the way you described it as being a junk drawer and just being not necessarily knowing the function is kind of very different to how the conversation has been described on YouTube because it being a battle between what's junk and what's not junk, and then the whole paradigm, what is creation, what is evolution. The evolutionists really pushed the narrative that junk DNA was basically non-functional, mainly non-functional to the highest degree, okay, and that it was just remnants from evolutionary past, okay, where you look at it like, well, it's not that functional, or maybe it could be functional, or whatever. So to
Right.
really draw the narrative, it has really been like, even like Dawkins said, you can basically get rid of 90 something percent of the DNA and nothing will happen. You know what I'm saying? And then a lot of the testing has been like, to prove the narrative that they've tested out these certain segments of junk DNA, and they were able to remove quite a bit. And, during knockout experiments or whatever. And,
Yeah.
right. And that kind of proved it wasn't junk. But it was a lot of misconceptions about that because, first of all, a mouse only lived like 3 years.
Yep.
So when you take away DNA from a mouse, you may, if it doesn't have a phenotypic effect on it, you may look at it like, okay, it's junk. But here's the problem. It's been a lot of neurological issues with the mice having this junk DNA taken away. And a lot of it didn't really necessarily show up because of his lifespan. And a lot of it was they had neurological effects where they were like stressed out, and they also had effects where Even though they did have offsprings, they had lower enzymatic action, and a lot of other effects that necessarily didn't show up, you know,
Yeah.
because
Yeah.
what it really boils down to is we do not have a good method as far as really framing what's junk and what's not junk. There's no good
Yeah.
method for that.
It's difficult. It's a difficult project. There are some things that we can determine, but it wouldn't be worded as junk and non-junk. We would call it conserved and non-conserved DNA. So either, like if we compared your genome to my genome, we'd find a lot of differences. Uh, on average, around 3 million base pairs would be different between just two random people in the United States. There'd be a lot
Yeah,
that much of a difference. So
right.
just by the way we're mixing. So
Right, but see, the thing about it is, and I think you're referencing some of those studies of recent where they found that it was more that even human by human, there is, if you do it telomere to telomere, that you could find
so
larger differences, and that was kind of a something they came out with because when they did uh the one study when they found out that it could be 15% difference from human to chimp, right?
yep,
You know,
yep,
so what they did was they looked at some human stuff and they found out there's a bigger gap than what they thought it was. But
yeah,
what they were looking at was from one spectrum to the next. If you look at like um so like Samoans compared to pygmy, yeah, you're gonna find a wide range there. But from the average human to the average human, it's very little difference. Very little difference. So that was kind of like to make up for the fact is that because if we're gonna be honest, evolution wants to prove that we are a common ancestor, and creation wants to prove it's not. And it's really going to determine how you look at the evidence. So if you don't really have any good methods for checking the DNA because you don't know which cell type is being what you don't necessarily know what these sequences are actually doing, because they're working in a they're being co-expressed in different cell tissues. And if you don't check the right tissue being tested, you won't come up with function or you'll come up with a low expression rate if it's not going to be expressed at this particular time anyway, because of the fact is that maybe it's just it's not being expressed, or maybe it's expressed one time or twice in a cycle, or during certain uh periods of development, it's not going to show a function. So we really don't have a good method. And I actually came
totally.
across something that said, how many reads, how many the methods they use and what they can actually test. And it's just quite insufficient. I wish I was looking for it, but I can't find it right now. But go ahead.
Yeah, I'm muted. Okay, we do have methods. We know the 200 tissue types, and we look at embryos as they're developing. Like we can take a frog or a chicken or a zebrafish, and we've very much detailed all through that animal's life what it comes up with. Now there are a few things like oh, long non-coding RNAs that might be important in a certain disease, but we'll get to that because that's a little nuanced. But as far as okay, in the genome, there are vast sections, okay, that where there are no genes. That would be like the telomeres, the centromeres, the alpha satellite on the end. There's big regions, desert regions in between here and there in the chromosomes, and there are no genes that we found. And yet, occasionally, some of these things will transcribe spuriously, but not not by any great quantity. So it really doesn't matter. But I wanted to show you So we can narrow it down and get down to just genes. Okay, what we call genes, what we mean by a gene is a gene that we have found out, that we have annotated, right? It doesn't mean that there isn't a gene there that we just haven't found yet, but we found a lot of them. You know, we've gone through testing all these different tissue types, and we've added up all of the mRNAs that we can find. So we got a pretty good map of what the genes are for any particular tissue type as we go along. And so when we get down to genes, let me make this big. Make Mike big again.
Okay, but can I say one thing really quick before you get too deep?
Yeah, go ahead.
So it's not just genes that has and that's why we really got to really understand, you know, genome function. Genome function could be that you have a long segment of DNA that's actually helping with transitional pausing. Okay, because there's some segments that had to take 8 hours to completely develop for that particular protein or that folding to be perfect. Okay, so you have segments there that really kind of like that go through the cycle for that proper fold. Then you also have quite a bit of segments that deals with chromatin structure. Quite a bit that deals with, communication between having a certain segment at a certain loci where the communication between those segments are in 3D space on a chromosome. So there's a lot of stuff. Go ahead.
You cross between two different entirely different domains. Just now when you were talking about, we have transcription within the nucleus, and then when it leaves, the mRNA gets out of the nucleus, then we have translation. So that's where you're going to have ribosomal pausing and folding and things like that. So we don't want to confuse that. We don't want to drag that in. What we're talking about in the nucleus right now, or specifically, is just transcription, where the DNA turns into some RNA, a sequence of RNA, right?
Yeah, but
for a particular gene.
What about communication too? Interaction, regulatory interaction though. And that's not necessarily transcribing into a protein, but that's regulatory reactions that has to be at a certain loci that works well too.
And that is another layer on top of what I want to talk about first. Okay, the first thing I want to talk about is this. Okay, so protein coding exons. Okay, protein coding. Let's see, total exons and introns for proteins in our genome is 25 to 26%. Okay, that's so if we took protein coding genes only out of our genome, that would be about 25%. Would be that. Out of that, most of it's introns, and I want to show you something just to illustrate. This is here is let me go up here a little bit. Okay, here's a good example gene here. What have we got? CREG2, and I'm showing you both. Let's see, I got to take.
Wait a minute, hold on. Wait a minute. Did we? Okay, so are you on the? Okay. That's a plus strand in the. That's on the right thing, right? Because I saw something at the bottom screen. We looking at the same thing, right? We looking at.
I think.
Strand plus 2, and one on the minus strand, one on the plus strand. That's what we looking at, right?
Right. Yeah.
Okay. I was making sure. I saw something on the bottom that I think it said creation myth. So I thought we was off the.
Yeah, I had that. Yeah, I had a comment up for a moment there. So what we've got here is I've got a little Python program where I can display both the chimp. So I've got chimp and human DNA, and I'm putting the chimp out for human out first, and then on the bottom I'm putting a chimp. So on the human we have this plus strand gene. No, this is a protein coding gene for PLEC. I don't know what the hell PLEC does. I haven't looked it up, but it
Right.
is about 34,000 nucleotides long. Okay. Now,
Okay.
out of that PLEC strand, and then in the bottom here is a chimpanzee, and this is chromosome 2, so they're reversed. In chromosome 2, half the genes are there'll be negative strand on the human and positive on the chimp, or vice versa. So just not to confuse it, but you can see the similarity between these two different things, right? So here we
Right.
go. So these are exons, these little tiny blue sections. So you can see how small that is. I made
Right.
the vertical bar high, right? And then on the end here, now this is exon 1 that you can see under my cursor. Exon
Hmm.
1, the coding region frequently doesn't start until halfway through or toward the end of that. So very little of this. So most of this is introns. And here's something we know about introns. Okay. And this amounts to about I think 25% of our genome is in the introns. My I have it over here somewhere. We'll go back to that. So what happens is on these introns is there's a little GT right here on one end, and that tells it to we got an intron, and then it'll find an AG on this end, right? But then in the middle, there's like a little kind of fuzzy sequence that it recognizes, and what it does is it pulls that up, and it goes over and it forms this lariat, and it's kind of like a little loop. So it's like you're roping a cow, okay? And then that gets snipped out, and these two exons will end up right together in the mRNA. But now what happens with that little lariat, that loop, that you know circular rope we have, is your nucleus recognizes that as junk that has to be degraded and recycled back into the mix. And so saying that that's functional. Now, that doesn't mean that sometimes there aren't little slices of this that end up becoming RNAs. We found a few of those, a small percentage of those. But for the most part, if you were to keep a mRNA for all of those introns, you would die in about 5 minutes because you would run out of the ability to create new proteins because all this has to be recycled. You'd end up with too much proteins. So we can make a damn good argument for all of this stuff in the middle, except for this fuzzy sequence and the ZTAG, that flag and intron. But that's the sequence of those genes are not important. And in fact, this is where you'll find all the lines and signs and ERVs, for the most part, are all inserted in this area. So what do you think of that? So I mean that is my argument, that there are sequences that don't do anything.
Okay. So like I say, I don't know. I don't. As far as I think what you're doing is you're talking about what these sequences are doing as far as protein production and regulatory. Okay. But these sequences could be, like I said, it could be having other functions. Now, we definitely know that there's a lot of the most the exons are protein coding. Okay, and just the fact is that the already polymerase or the spliceosome is not grabbing that kind of speaks to um, you know, specificity, So from an evolutionary standpoint, okay, why would, because you called it junk. You said it's junk, and that the spliceosome is ignoring that because if it did too many transcriptions, okay, you would never produce any protein because it's doing too much work. That's what you said, right?
Well, basically, we use a lot more. We use a lot more nucleic acids in the nucleus, and I think we'd run out very quickly because that would be too much. Our job is to produce these proteins, right? We don't want to
Right.
We don't want to
right.
keep all this other stuff around. But what you're saying is now you're going to go to a different step outside of genes, and we're going to talk about regulatory and
Right, and
chromatin
right,
remodeling. Right.
right. So I agree with you on that because they're not, the everybody know the introns are not producing proteins. There are some though because you got dual coding genes where you have I think it's called, you got duons, you got dual coding genes where, because it used to be, you know in evolution, and tell me this isn't true, one gene, one function, right? Now they're finding dual coding genes. Then of course, they really learned about the alternative splicing and all that type of stuff. That they didn't really talk about. But you have, just because those introns are not producing a protein, it doesn't mean it's not serving a regulatory function. Like with the things I said, the chromatin building, the cell signaling, interreactions with TADs, epigenetic factors, and stuff like that. So I think that if you just want to call junk DNA something that is not producing a protein, then yeah, if you want to call that junk DNA, then that's junk DNA. But it's much more than that.
Well, not all the time. It doesn't necessarily have to be. If we haven't found a function for it, we can't
But
just assume that there is a function. Because if there
right,
was, why would you and I be so different? Why would we have that 3 million differences? That would mean you'd survive and I wouldn't.
no, like I said, it deals, and I don't think that we're that different. I think that that if you're talking about if you're talking about 3 billion letters, okay, even if I'm not sure about the 3 million. I'd have to do my research on that one. I don't think that person by person. I think that if you're looking at one totally side of the spectrum, as far as you're looking at a pygmy versus a Samoan or something like that, yeah, you're going to see that large spectrum of change. Okay, but I don't think that we're the average person is 3 million different. I don't really believe that, to be honest with you,
That's
because they
kind of
didn't
what
even
I
talk about that. So let me just say one thing. They didn't talk about that. Nobody really mentioned anything about that until they found out that when you do a telomere to telomere sequencing, right, they were
Mhm.
expecting to see more similar to chimps. When it wasn't similar to chimps, when it was like 15% difference, then they started talking about,
No,
well,
it was just the opposite. Just the opposite. When we did a telomere to telomere sequencing between chimps and humans, we found more similarity. I did anyway in my research. I found more
yeah,
similarity than before
but you wait a minute, you found more similarity. Well, it's
to
what
chimps.
they saying it's 15. They saying it's 15%. See, and
Okay,
it really depends, right? And before, uh,
let me
we'd be
complain.
lucky. Hold on, let me just say one thing. Before, we'd be lucky to get away with 5%, because even in I think it was a 2005 paper, when they looked at it, they said like 5%, but they didn't look at the whole genome. It was a bunch they didn't look at. So,
Right.
so
If I
now
look at
it's like,
my
go ahead.
calculations with just genes, I found something like zero point 0.8 to 1.2% differences in all the genes that I came up with. And out of those differences, almost everyone were synonymous mutations, so that they wouldn't have made a difference to the protein at all. Uh, with
Well,
it,
you don't
like
know.
I think
Okay,
it looked like 7 out of 10.
okay, so you don't necessarily know that because if you're
I know
talking
that. I
about,
can look at the genome.
okay, but let me address that real quickly. When you talk about
Hmm.
synonymous mutation, it doesn't change the amino acid, right? Because that could be the third position wobble, whatever. Okay, but if you look
Right.
at, but we don't know. We don't know since you got a lot of overlapping in genes. Okay, so and you got sharing you got these sharing these same sequences. So if that amino acid could change, okay, that amino acid can change that protein, but we don't know if it could be a certain a little bit weaker in transcription or regulation. You don't know that for certain. You don't know because if let's say if you can produce, you could produce in one particular segment, it could be like uh, four overlapping genes. So one gene, no effect, but you don't know what those other genes could be. You don't know how the regulation could change because we're not sophisticated enough to be able to make those distinctions. When you got such a huge genome, right? And you got all these interreactions where it was once said that the the gene, the DDX11L2 gene, that that there was very little transcription in that gene. But then when you looked at websites that kind of focus on gene associations and co-expressions, then they found out there's a ton of co-expressions. There's a ton of other genes that's
Oh no. DDX11L2. Okay, that's the chromosome 2 stuff. No.
no 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 no
DDX11L2. No,
no
nothing.
no
There's no function.
okay so okay, so hold on for a minute. Let me see if
No,
I can find.
let's not go down that rabbit hole. You don't want me
Okay,
to go
okay,
down
okay,
that
okay,
rabbit
okay,
hole.
okay. Let's not use that example. Because yeah, we will
Okay.
be. Yeah, let's not use that example. But what I'm saying is, since they're kind of focusing on genes as far as what they do, as far as with God, I'm so sleep I can hardly think. But what genes do with as far as health is concerned, um
Yeah.
you know, they're focusing on what the junk DNA do because it has a strong effect on the health. It has a strong effect on how we live. Okay, and I meant to say disease. My brain is not thinking right now. So when you look at the effect of junk DNA in genes, they have to look at it now because it was so much emphasis was put on what protein genes doing. Now they have 90 something percent that's doing regulatory, that has some type of a regulatory effect or something. The introns and the non-coding genes and all this so-called junk having an effect in disease. Now you're trying to figure out what they're doing, and they just don't know. It's just they don't know because they don't have any good methods, because they spent so many years trying to figure out what protein coding genes do. Now they're so far behind, and it's such a larger portion of the genome that's involved with regulation and disease. So you just can't say anymore that this gene is junk, or we even got to change the word junk, to purpose. Is it serving some purpose?
Does it do something? Okay. Like
Right? Does it do something?
every single nucleotide in your genome is an actual molecule linked together into one big molecule, and it all does something. It's all going to have some effect. It might waste space. It might cause some heat. I mean, yeah, there are a lot of complicated layers. Okay, but you're saying something a little bit different about this now, and I just want to, so I want to try to get down to what you're saying. So we're talking about this intron here, right? We got an intron right here. Okay.
Mhm.
And that intron has a certain sequence, it'll be T, C, A, G, 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 whatever. Okay. What are you saying, that intron was designed so that it has to be exact? It has to be that particular sequence. Otherwise, what?
No, I'm not going to say exact because a lot of them is exact. But the thing about it is we have accumulated some mutations too. But
Okay, you're saying a lot of the introns have to be exact? What are you basing that on?
well, okay, so okay, I'm not going to say exact. I'm going to say that there has to be is very beneficial for it to be the redundancy that we see. The redundancy is really important with the
Right.
chromatin structure, with, like we said, the transitional pausing, the interreactions between even self-signaling. You know, those locations are important because you got long range and short range. It's just like it's like a I guess you can consider it like a operating system, you know, random access memory. Okay, you want to have that spacing there. You want to have where they can communicate back and forth to be efficient. So, you know, if you look at it in that light, it kind of changes the whole, you know, perspective on junk DNA.
Okay. So 98% of this thing is introns, right? 98% of this one gene is introns. And this is just one gene. And out here, we've got a regulatory sequence out here, right? So there's a little bit of regulation, or probably a vast region of regulation. Oh, you can't see it. Oh, that's too bad. It's there anyway. Put it down, right? So 98% of this is introns. So what do you say? So you're saying that 98% that we had blinkers on, we never looked at it, we didn't care about it, and that's why there's this idea that biologists had it in their mind that it was all junk DNA, so they didn't look. Well, I got to ask you, CBrown, if we didn't look, then how come we found it? How come we
I mean,
found some function?
you're gonna find it later. That's just part of science. You're gonna
Yeah,
focus on one thing, and then you make a discovery. Oh, wait a minute. And then
right.
you
Yeah,
see more, and you
exactly.
see more, and you see more.
More and more. Right, right.
Right,
Okay.
right. But if
So
you
anyway,
start with the perspective, hold on real quickly. If you start with the perspective that,
yep.
you know, this is junk and this is evolutionary remnants of the evolutionary past, and that it had a function before. Now it don't. If you start with that, you know, and then you have to find function of 98% when you thought it was only 2%, it's going to be pretty tricky because it's um
It
it's
is
hard to
tricky.
determine what noise is. You know, it's like say you got a whole bunch of things going on. How do you determine what is actually what this set of sequences are doing? Where is it coming from? It's hard to
Okay.
figure out.
There's one way that I can figure it out. Okay. We've taken thousands of human genomes, right, and compared them. And what we'll find is that this part right here, this exon, we'll find that it's highly conserved. We'll find that this intron, not highly conserved. Doesn't matter if you have a mutation, I have a mutation, we're both going to live till we're 80 or 90. Not going to make any difference. There might be some weird little health effect. We might get like sepsis, and one of us will do better than the other. But then you've got to add up the entire genome, and it's going to determine the health of the individual. But that's a holistic thing, right? You can't go down and tell me that you know or that you're going to find that each and every one of these nucleotides is absolutely important. And here's why: because mutations are common, because we're firing bullets at this all through our lives. Okay, the gamma rays from the sun, smoking a cigarette, drinking some vodka, we're firing bullets, right? And if you have this big long sequence like this right here, the chances are that the bullets are going to hit some junk. It ain't going to matter. It ain't going to matter what happens in mutations to this, right? So that's the important part. But now think about it now. Maybe God designed it that way. Maybe God put this buffer in there so the bullets wouldn't hit. The important part. Here's the important part. If you're I'm trying to think of an analogy, like with a shootout, if you're running down the road with, a little tiny car, and there's a whole bunch of other cars, and you don't know which car the guy you want to kill is in, and you just start shooting, you're not likely to hit that guy, right? So it's not likely to make a difference. So why couldn't God have designed some buffer here for us in these introns?
Okay, so wait a minute. I'm trying to understand. So you're saying
Hmm.
that God could have designed it as junk. Is that what you're saying?
Yeah, yeah, God could have.
Okay.
Yeah,
So yeah. Okay.
God can do
So
anything.
I don't know if I believe. I don't know if I don't agree with that. I think that
You don't? You don't?
no. I think that
Why?
because okay. So for one thing, redundancy isn't junk. Okay, redundancy is backup mechanisms, right? So I don't necessarily agree with that. I think that if we look
Well,
at the whole totality, so what we have to do is we have to look at two. We got to broaden the picture out a little bit. We got to broaden out. We got to say what makes more sense. That all of this is junk from random, past relationships between what we used to be. Okay, to is this created very super complex, right? Now we're finding these new functions and we're finding that there's just so many different interactions and different layers of the genome. It's just more or less intelligently designed. And it's just designed to where we don't really know exactly what it's doing. It's like, okay, so you remember when we talked about like 95% of the spliceosomes, the transcripts that it produce are tissue specific, and the spliceosome being extremely accurate, as snipping out the right, you know, uh, exons for the particular proteins. That speaks to a high level of intelligence. It seems like your narrative is we just get lucky in certain times, and the rest of it's just stuff we just don't need.
I don't
And
use
and,
luck.
and
I
that's
don't
how
do
we.
that.
Okay, so why do you think the spliceosome is tissue specific? That means that if it's tissue specific, that means that it's grabbing these introns, exons, okay, for a specific tissue, okay, that is deliberately ignoring specific exons that is not needed in this transcript. So
Okay,
that's intelligence.
you're
And
talking
that's
about
and that's
alternative splicing, right?
alternative splicing, yeah. What
Yeah.
this what the spliceosomes are doing. Period.
Yeah. Yeah. It looks like intelligence to you, but it doesn't to me because basically these exon. Well, now we're going to get down another little rabbit hole, which is proteins, right? Which how proteins work.
Well,
These
it ain't just
exons
protein.
often
Go ahead. Go ahead. I'm sorry. Go
these
ahead.
ex these exons are often they have things like they're called domains. Like this right here might be the functional domain of the protein. It might be what hooks up to a particular little, say, some random vitamin or sugar molecule. Okay, this right here, and this right here maybe does nothing, but these two things right here, they lock onto a particular protein in a complex, right? And now, okay, we keep this part, right? So this part gets transcribed, okay, but this changes right here, so only this part gets transcribed. So now we have an alternate protein, and now we have one protein that's going to hook up to A and one protein that's going to hook up to B, right? Okay. And in evolutionary history, all we needed was A. But now we've added this new function, B, for alternative splicing. So it can happen incrementally because of the way proteins work. But I want to get back to introns.
But that
Okay,
sounds like luck. That sounds like luck, though, to me. I mean, that really sounds like luck.
it sounds like change to me. Like if I look at my oak tree out back, I see that on the one side it has this really thick branch, on the other side it has two thinner branches. You want to call that luck? I call it causality.
But
It just
okay,
turned out that
okay,
way.
okay. But what tells the spliceosome to take a notebook to continuously grab these exons? So the thing is the spliceosome is extremely accurate. Okay, it's grabbing the right exons, and then it's taking a notebook and it's continuously doing it. Then it's continuously grabbing these other ones. It's continuously grabbing these other ones. Why does it have to select the correct ones all the time? And then you got a system that the ones that don't correct, it degrades. And then you also have um correction mechanisms that okay, if this is not the correct one, let's destroy it. So that's a lot of luck to me. That sounds like a lot of luck. That it sounds very specific. This is what we want. This is what we don't want. Okay, this protein is not going to fold correctly to interact with these other proteins. Let's destroy it.
Okay. There's a lot to unpack there, and I'm not even sure which way to go. But anyway, okay. So I told you about the alternative splicing, right? I wanted to get back to introns, but we're not going to. So okay, so we have organism A, and organism A has oh my God. So I used A twice. Organism 1 and organism 2. Okay. So organism 1 has a liver that basically processes some chemical in the environment. Okay. And it needs A to do that with, right? And then time passes, basically. So, these things, mommy and daddy love each other very much, and they keep moving on and reproducing, right? And they eventually create this. We have this mutation here where in the liver tissue, particularly, okay, the spliceosome is such that we have both A proteins and B proteins, right? Okay. Now this new oh, did you put that up there, Scott? Because that's the things that work and work. Okay, I'm gonna set that aside for just a minute, okay?
Right, because that's
Okay,
not a good point.
so now we have organism B, okay, which can process both A and B, and it runs into like these two organisms, okay, these two different lines, two different species. One ends up on one side of the river and the other on another side of the river. And on this side of the river, it just so happens that A and B will kill it unless it processes both these things, right? On this side of the river, just A will kill it, right? So now we have a speciation event. So that's how it happens. It has nothing to do with luck. It's just these two populations. They ended up with a mutation here. It spread to a certain part of the population. The ones that went down here, everybody who couldn't process B died. So now we have a new thing. So that's what I'm saying. That's how this works.
Yeah, I don't.
So
Yeah,
so I'm looking in fine detail at everything that happened to this genome and how it developed. That's how we look at it in evolution. So I don't see where you get luck out of that. I don't see luck.
I don't see natural selection being that sharp that where because we're talking about before selection. We're talking about like when you got error correcting protocols. Okay, error correcting protocols mean that something has to be specific. If it's not, it's got to go. That it seemed like it's some type of forward thinking there. Okay, so this happens before natural selection takes them out. Okay, so natural select, this is the after effect. But the whole thing is when you do make a mutation or you do select these introns or whatever exons, why is it selecting these? Why is it skipping this one for this particular protein, but then it's skipping the other ones for this particular different protein, and they all work. Okay. And if it didn't work, okay, if it wasn't correct, it gets destroyed for food for the cell.
Okay.
Evolution shouldn't be taking a notebook. It should be everything should be random. It shouldn't be that um we're building this one, then we're building this protein. The same way you have, like, I don't mean to get too far into it. No, actually, I don't want to get into that. You can answer it because it will be way down away from where we want to be. But go ahead.
Okay, so because Deconverted Man said because things that work work and things that do not work don't work. That's natural selection. It's like asking why 1 + 1 = 2. It just does. There is no why. Carbon is a slut that loves up to 4 things and connects to them. So anyway,
Yeah, but
that's
things don't really work random like that, though. That's the thing.
they work well.
When things work, that's
Okay,
because when things work like that, it's because it was designed to work. Because again, you have safety mechanisms there. You have regulation, and you have quality control. You don't get quality control from randomness.
why
They don't
not?
do quality control. Why would they? Why would they even recognize and know what's right? You know?
Because okay, if you have a factory and you have a quality control, you hire 6 quality control inspectors, and 3 of the quality control inspectors put out defective product every day. You fire them, they die. Now you have 3 quality control inspectors that are better at their job. That's how natural selection works. It's the
No,
quality
okay,
control inspectors that don't work are going to get fired, right? So
but here's the thing, though. The difference is, here's the thing. These quality control inspectors, they are intelligent. So they are. If the only way it's not going to work if they're not following the instructions. Okay, if they're not following instructions, of course, you know.
no, what if they're
uh,
just what if it depends on their skill level? They're just random stuff they learn throughout their life when they come to the job. Some of them are really good at this job, and some aren't good at this job. It isn't about
well,
reading instructions.
well,
There's no
yeah, but that's
instructions. They're just
yeah,
good at it or bad at it.
but that's kind of different, because I don't think that evolution is as smart to know what's going to work. It's going to work. If it ain't going to work, okay. If it's not going to work, then
They die. They
it's
die.
going to
It's
dump.
that
No, no,
simple.
no, 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 no. It can go a very, very, very long time with it not working. But the main problem is this here. It has to keep copying the same thing over and over again. It's got to know. And if it don't know, then that particular one is gone. The system is being upgraded constantly. It's being checked
Y-
constantly.
Yes. Yes, it
E-
is. Constantly.
Evolution can't do that.
Yeah, it
It don't.
can. Natural selection can do that. It's just
So
it's
you
a platitude that what doesn't work isn't going to work.
no.
Whatever
So are you
doesn't
saying
work dies.
we would? And that's the reason. And I guess that's good. I hate to go here, but I guess that's good. That brings us to genetic entropy. You know, that's what it brings us to. So okay, let me
Yes.
ask you a question. Let me ask you a question then.
Yep.
These master proteins that can basically regulate hundreds and thousands of genes. You know, where they come from? I mean, that's a lot of levels of complexity. Because it really boils
Mass.
down to is this complex? Is this purpose, or is this just random chance? That's just
We don't
get
we don't
it.
have hundreds of thousands of genes. So,
Okay, okay, okay.
uh,
I'm sorry. Okay, okay, okay. Not 100,000 genes. Okay, you're right. Thousands of genes. I didn't mean to say 100 because I think it's like 20 something thousand genes,
we
something
got
like
about
that.
we got about 20. Yeah, we got about 20,000 genes. So what are you talking about? Does what to what?
Right. No, yeah, that was a misspeak. I told you I'm
Okay.
sleepy right now. I should have
Okay,
uh
that's all right.
right.
Yep. So
So
let's let's get. God, I don't know if we should go in the natural selection route or go back to our introns. But okay, so we're gonna end up with right here, basically. what we have is right here, we have this random mutation where it starts splicing, this alternative splicing in your liver, and it can handle this B product, right? And out here, it does not matter, basically, okay? But somewhere along the line in the future, okay, there's a river that basically divides these two species up, and now we've got some out here. We have A types, and we got a few AB types who can do both, and an A type and an AB type, right? So we have both those things, okay? And somewhere along the line out here, the environment changes so that there's a lot of B in the environment that can kill you, and your liver needs to take it out of the environment, right? So what's going to happen is in this population, you're going to end up with a mix. At some point in the future, that B type might just totally disappear because it's not being used. It might drift out of the population. But now down here, all of these A guys are dead. They're all going to die right here. So now we're going to have the AB gene. So now we have two different species, one that can handle AB or subspecies, and one that can handle just A, because this eventually might just
Okay, let me ask you a question though. So what
Okay.
about the fact is that and now you're kind of getting into epigenetics, right?
Oh no, that's a whole nother talk, and
Okay, well, in
we're
a
not ready
sense,
for
yes.
that.
In a sense, yes, because you talk about the environment. What about the natural ability to adapt to the environment?
Oh
So
no, we're
you
not
can have.
talking. Okay, wait a minute. This
So
is
I'm
not
saying that might not
epigenetics.
be.
That's
That might
not.
not be. What I'm saying is that might not be the result, though. Is what I'm saying. That might not be the
What do you mean?
result. Okay,
Why
because you're
not?
saying
Why?
that you're saying populations die off, right? This
Right.
population die off, right? So what if instead of dying off, that the immune system, the epigenetic system, make a change, okay, or it express a certain, it shut one of these down. That's not beneficial
Oh,
for the environment, but you still have it, but it goes dormant.
yeah,
So I mean
yeah,
that's
yeah. So in the future, okay, basically this could change so that B only happened because of an epigenetic modification, right?
right.
So that could happen, and that would be sort of a random thing, right? So now,
Mhm.
what would happen is that epigenetic modification got turned on by the environment, right? And
Mhm.
that would be just a modification, basically. So that would mean that the B protein that we got wasted, this organism was a little more efficient because it didn't waste its time making B unless it needed B. So I mean, that's just a small change, that epigenetic modification.
Right.
So I mean, I could pop in too, but we don't
Yeah,
want to
but
go down rabbit holes that we aren't ready for. So
right. No, I won't go to rabbit hole, but I will say that here, epigenetics is that's another level. That's another level of complexity. So at the
yeah,
end of the day, at the end of the day, we have to say that evolution, main tenets, it operates on randomness, and then what's
your
beneficial.
mic just went. Something just happened to your mic. Are you too close, too far?
Oh,
Try
I don't
again.
know. Is it? Can you hear me? Wait a minute. Can you
Yeah,
hear
it's
me?
kind of. It,
Okay,
yeah, it
hold on.
really
Let me
went
try.
bad.
Let me try to put the earpiece in and see if I can. Oh man, I hope I don't click this off. Wait a minute.
Okay.
All right. I'm scared to do this because I may ended up. Hold on, man. I hope I don't do that. Let me see. Mike. Okay.
Yeah, it just suddenly went bad. So something
Okay.
changed.
Okay. You can hear me now.
No,
Can
same
you hear?
thing.
Oh
You're
man.
it's yeah. It sounds like you're vibrating, like you're talking through a flute or something, a flute mouthpiece,
Okay.
vibrating like.
All right. Hold on for a minute. Let me try to turn that off again. Let me see.
Yeah, Mike,
Maybe
I think
try a
that's on
different.
your end. I don't hear
Oh, it
it.
is.
I have TP Seeker here saying it sounds good to them.
Oh, you're doing it too. Oh my God. So something went the hell on my end.
Oh, okay. You made me change it for nothing then. Okay.
I
I was
did. I'm
sounding okay.
sorry.
All right, hold on
Okay.
for a minute.
Mike should
Okay,
have
I
been more
can understand.
scientific.
Yeah, what is going on? Oh my goodness.
Right. So I sound okay to everybody else?
Yeah, I guess so.
Okay.
And I still sound okay. I'm not
Right.
I'm not modulating.
Okay.
What in the
No, you
hell
sound okay to
just
me.
happened?
Yeah, that's no problem.
Yeah.
Okay, but okay, now
Shit.
somebody said in the comment that I gotta be schooled. I'm only on level 1 of population genetics.
Popped
You
in.
know.
Yeah, let's just clarify that for just a moment here. Okay,
Mhm.
in my diagram here, when I was talking about these things, okay, I'm
Right.
talking about a whole population, like maybe 100,000 little beetles, and 100,000 little beetles here, and 90,000 have this modification, and only 10,000 have this modification. And what's going to happen is we're going to lose 90% of the population, and these are the only guys that are going to survive. But it's not
Yeah,
just one organism. We
right.
we
But that's
don't want to
kind of
mistake this with one organism. I'm gonna have to get out of the studio and get back in, guys, because something went wrong. So
right.
Scott, can you come in for a moment and
I am
Dr.
in for the
CBrown?
moment.
Okay,
Okay, I'm
so
gonna try to get out.
I want to
I'm gonna get
welcome.
out and
I
get.
want to welcome Vagda to the
I'm getting
side
out.
chat. Vagda is actually Demeter, who used to be 4th or 11th dimension, I believe. I can't remember the entire handle. Deconverted Man is in the side chat, and I think Dr. Dan might have gone off to have some Thanksgiving dinner because he's stopped commenting, but he's been commenting.
right. So you know what? As far as that diagram that he wrote, I think that particular thing, yeah, that means I guess you can get creatures to go extinct, but that's something that kind of really don't happen too often. And it's I think that's something that's when it's like an extreme change. So you know, I agree that mutations can do certain slight changes or whatever. My whole thing is, you know, common ancestry. What he said, I agree. I basically agree with that, but I also agree that that could be the flip side of that could be that it could be a program mutation too to help adapt to the environment. You know, so I don't see that as being evidence for uh, against junk DNA being against my case being as that junk DNA is not as prevalent as everybody think it is.
Yeah, I don't want to call it junk DNA. So I basically, it's all of this stuff is important, even if it's just to absorb bullets, right? That like gamma rays, or the thing we have here are signs and lines. Like if you look at now, I could show you, but it would take me a minute. But I could show you in these introns, they are filthy with viruses that have popped in, that have jumped in, and they don't make any difference. We survive just the same because what they do is they just keep expanding this intron and making it bigger, and these things don't matter. And we can tell they don't matter because if you look at basically two different humans, you're going to find a lot of differences. And if we compare humans and chimps, we can tell they don't matter because they're going to have even more differences, that sort of thing. So, I mean, that's so, like I say, we want to get away from this word junk DNA. If anything, we'll call it junk drawer DNA or
Right.
utility
Okay.
DNA. Yeah,
Well,
whatever.
that's kind of honestly, that's kind of
But
one of the most honest things I've heard, honestly, because you know they
that does not give you design. That does not give you design.
I think it
So,
do. So it
well,
depends. it really depends. Again, it really depends on how you it's the starting point. You see things as, evolution, points to evolution, no matter what. No matter what, it points to evolution. Every time but here's the thing: the more complexity we find, that to me definitely. Oh, am I gone? I think no, wait.
No,
Oh,
you're fine.
I clicked out on this other one. Hold on for a minute.
You're fine. I can hear you.
Right, but I think I'm clicked out. Well, I'm not. I can't see the screen on the other one. That's okay. I ain't gonna worry about it. So I just, I think the way we got to argue is what is intelligently designed and what came about through the evolutionary system. But I think the evolutionary system has to rely on randomness, and I don't think that randomness can get you to extremely complex design.
Well, it can't be considered random except from certain points of view, right? The word random is something we humans use to when like if we look at an avalanche, right, and we see all these rocks, and the avalanche is over. It happened at 7 a.m. this morning. Now it's over, and we go out and there's all these rocks in my yard, and I say, oh, they're randomly arranged. That only means that I don't know what they bumped into on the way down, right? So the word random is only used in statistics, and it's the point is that for a particular mutation in the DNA, unless we go down and look at every little atom bumping into every other atom, we're not going to be able to predict which mutation is going to happen. And then we'll say, oh, that's random. Okay, so that's what randomness is. But what you're talking about is something else. When we're talking
Yeah,
about evolution,
right. But it has to be random if it's not purpose. If it's not, if now, unless you want to say that God is involved, it's got to either be random or it's got to be purpose. I mean, you kind of can't have it both ways. Yeah, you got to. I mean, it's because you have to set up a whole system in order for everything to operate out of. So you would have to say the whole system just came from randomness, and then that means that it got lucky lots and lots and lots of times. I mean, I don't know how else you can.
It
Go
doesn't
ahead.
it doesn't have to get lucky to the degree that you think it does. That's what you're missing. Okay, like we got these like each of these are nucleotides, right? And they have like little spinners, that we spin. I'm drawing something. If you can't see it, I'm hoping you can
No,
see it.
I can't. Hold on. I can try to hold on. Let me try to get back into that. You know what?
Okay.
My mouse. Oh, here we go. Let me see if I can get back into that screen where I can see that. Wait a minute. Is this screen right here? Hold on a minute. I just want to keep it muted though, so I can't. All right, let's continue back in here. Hopefully, I'm still muted. Okay, stay muted. Can't find microphone. Enter without microphone. Okay. Am I?
Okay.
Okay, so I'm not echoing, am I?
No, you're fine.
Okay. Yeah. Okay, so I can look at your thing again.
Okay, so we've got these little spinners, right? And they can land in one of four quadrants. That would be, A, C, T, G, right?
Right.
From wherever it is, right? Say it was a G and it can be one of those four things. Yeah, we're going to call that random, right? But the thing is that doesn't matter so much because as it turns out, we're still going to create, this little protein that does what it does and it works, right? And it doesn't matter. It doesn't matter that any one of these things change, right? But if three or four of them change, then this little protein is going to start to have a slightly different structure, right? And that little protein is either going to work just the same as it did before, it's going to work better, or it's going to work worse, right? And if it works worse, we've got a dead organism, right? It's going to die before the embryo is before before the lady even knows that she is pregnant, it's going to die, right? Because a lot of like 40% of the fertilizations, the mom and dad don't even know that somebody got pregnant because there are enough mutations that it died before it got even developed, that sort of thing. So I mean, that's going to pop out. And these things, okay. And then so we have now what you want to talk about is this massive complexity that we have in a single cell. We have all of these things in the single cell, just many different things, right? And you got all these many different things, and each one of them just changes, or one or two of them might change a little bit from one generation to the next, and it just keeps going on. But overall, this whole thing either functions or it doesn't. And if it functions well, okay, basically it's more likely to take over a population. If it functions the same as some other change, the chances are you're going to end up with two different variations in a population. And that's why you need a big population in order to survive, because you got all these variations laying around. You got all this junk drawer DNA laying around, ready to go to work when like I showed you for that A and B example back here, right? So, and I guess you would call that lucky if you happen to have the protein that is going to help you survive when the environment changes, then you're going to luck out and you're going to live, right? So I mean, this complexity, these mutations happen rather slowly from generation to generation, okay? And they very slowly kind of move the population. And then what we really need is a large population that has a lot of diversity, which humans have right now, right? And if something happens, something changes, that some of the members of that large, diverse population are going to survive and the other ones aren't. And you're going to have a change, and you're going to keep changing. And when it comes to the liver, one metabolic pathway after another was added over tens of millions of years to end up with our liver that can, I think most of us can handle alcohol, but some of us can't, that sort of thing. So, I mean, the complexity is what causes it to, it's basically, it's what makes it not about random, right? Because there are so many different pathways that could have worked in this huge sea of even more pathways that don't work. And all the pathways that don't work are going to get stamped out at every step of the way.
Yeah, but the thing about it is this here, first, see, you're starting with full function off rip. You're starting with the full function, but I don't want to get off of that.
Yep.
The thing is what really kills that is there are active mechanisms, error correcting protocols that recognize and identify if something did not fold correctly or there's a bad mutation. There's even DNA repair mechanisms that repair the actual DNA. It knows if there's an error correcting on the DNA. Then what really makes things really bad is the topo. I can't get that word together. The
topoisomerase.
Right. The way that thing. How do you evolve something that totally unravels the DNA? Unravels DNA. So are you saying that some creatures did not, some species did not develop that to where it's unraveling the DNA so that you can even.
Yep.
Okay. So, wow. Okay.
Basically single-celled organisms that had very, very small DNA segments or probably RNA segments, were what we had in the very beginning. But I mean, like we're moving the goalposts all over the damn place now. Now we're going back to the biogenesis of early protein families, right?
Well, the reason why we kind of doing that, the reason why we're kind of doing that is because, if it's going to be a thing to
Because
where, wait a minute,
you're
hold
a
on, come up here real
creationist.
quickly. If no, well, if it's going to be if it's going to be a thing to where anything can happen, right? Any mutation can happen. These bitty steps could move it in this direction. You don't need a notebook to copy. Keep copying these same segments. Keep going to the right. Um, it keep selecting the right exons. It keep doing this, but you're not really saying how it's doing it. Then I got to pull back a little bit and say, okay, if you want to keep insisting that, then I have to look at the whole picture, because if I look at the whole picture, then you got more to explain.
Oh yeah,
Because,
we have some explaining to do. There's no doubt
right,
about it. That's what I do every day. That's the work that I do, is try to
right.
explain the evolutionary steps, right? That's what
Okay,
all
what
evolutionary biologists do.
but at what point do we say, wow, we just can't keep saying this is, if we find like three more layers of information, you know, um, can we keep saying that this happened randomly?
Yeah, yeah, of
As,
course.
as,
The more layers we find, the more likely it is. There are
so
more
that means it can't.
pathways. There are
That
more
means it can't.
pathways if it's more complex.
Well, that means it can't be falsified then, because we start with the premise. We start with the premise that it, it it's too intricately designed to be natural. We start with that. So and complexity is our friend, where you got to
Yes,
keep explaining it, you know,
right.
because everything looks the genome looks so designed, it's ridiculous. I mean,
It doesn't. It looks horribly undesigned to me. The more I look, but anyway, I got a lot to explain, and what would falsify it? I guess if I found, what the hell would it be? I guess if we discovered that the protein space, was that there was like huge sections around every functional protein where it wasn't functional, where it couldn't function at all, right? Then I would say, oh my God, then we got a
well,
problem. You got to
yeah.
figure that out.
I mean, well,
Or if
that's nothing to figure out. If that was the case,
if
we
I
wouldn't even
found it
have life.
too
We wouldn't even have
right,
life if that's the case.
right. So I mean, it has to be. So basically, but that's not what we're finding. What we're finding is that the protein space is just filthy with functionality, and the functionality is one or two small steps between each functional protein. So we've got this like complex where we can go this way and that way and this way and whatever. And
You
I
talk
mean,
about
there's just
you mean kind of like an operating system, right?
well, no, an operating system is pretty rigid. If I type one wrong character, my Linux command said, I don't know what the fuck you're talking about, right? It says,
Well, it
get out
depends
of
on
here,
where it depends
right?
on where you it depends on where it's at. If it's in the what do you call it, the red, I don't, it's still called the registry. They still call it the registry or the
Yeah,
BIOS or
well,
something like that.
on Windows they have a registry and a BIOS. They have both,
Right.
on
Well, if it's
Windows. So.
considered that, yeah. But I'm just saying, if it's
Yeah.
but you have to build the BIOS have to be, it has to be built by
Oh, look at
intelligence.
this.
But you can
Look what we
you can
got
have
here. See, one character, right? Watch what happens. Okay, that's code. That's what code does, right? It doesn't do not. It broke with one character. Okay,
yeah, but it don't
go
break the
back
whole
to it.
system. But it's
It
probably
broke the whole.
it's probably
It didn't
more
work.
lines of code. It's probably more lines of code there, But
There's
and that's what mutations does. So that doesn't help you. That's what mutation.
no
But
mutations
but you
don't work
want
that way. I basically if Python was like the genome, I would be able to make all kinds of changes here. I'd be able to put an equals there and a comma over here. I'd be able to do all kinds of stuff, and it still give me my picture,
okay.
right? And then
Let's look at it like the go ahead. Well, let's look at let's look at a football team. Okay, let's look at a football team. You know, you got quarterback, you got wide receivers. These guys is the guys that score the points, right? And running backs,
I
right?
don't
But
know what any
outside
of
of
those
that,
things are, but
yeah, you do.
no,
But
I
outside,
don't. I really don't. I've
okay,
heard the words before, but I don't know what the hell they are. A
okay,
running
so
back? Does he run backwards? Does he go back the other way? What does a running
no,
back do?
come
He
on
take the
now,
ball
come
the
on,
other
man.
way?
Come on,
No,
man. You
really, I don't know what a running back does.
okay? Okay, okay. So you don't
Anyway,
never you never watch football for real?
never, no, never,
Oh well,
ever,
I can't use it.
ever since I've been a little tiny kid. Yeah, I've hated it. Yeah,
Okay, well
they used
I
to.
can't use that
Yeah,
example. What I'm saying,
no, there's
what I'm
different.
trying to say is you got certain players that score the big points, but behind an organization, there's a lot of people that work behind the scenes, you
Oh
know,
yeah,
in different capacities. Different capacities. You lose one, okay, fine. It's gonna still work because you got backups, you got replacements, you got redundancy. And that's how the genome is. It's built with redundancy, you know. So
yes.
that's an intelligent
And
design.
and you know what that redundancy is? Junk DNA. That's a
Uh,
redundancy.
but see, it
Yes.
has to right, but it has to start. But see, but
Like
here's the thing though.
you have some pathways, biochemical pathways in all of our bodies that there are like one, two, or three different ways that work. And if one of them drops out, we're still going to survive. So it still happens.
Yeah.
That is redundancy. Okay. And that's because of this random, well, no, I hate, there I use the word I hate. So that's because of all of this. That's because of all of these pathways and this whole pile of forks and knives and junk stuff that we got in that junk drawer. Basically, one of them is going to work for us, and that's how we managed to stay alive for 100 years. Otherwise, we die. But anyway, we're burning out here. I'm at an
Right.
hour and a half. Yeah,
Yeah, I'm tired
hour
too.
40.
Yeah,
Yeah,
I'm
okay,
ready.
yeah. And it's
But it was
Thanksgiving for
something you
Christ's sake.
said. It was something you said that I meant to take notes on that I really wanted to study that. I forgot what it was. I should have wrote that down because I
Well,
wanted to something you
if
said
you think
way
of it,
early.
if you think of it, let me know. I can also sit on with you privately and try to tell you some of the stuff I know. And I don't know a hell of a lot, but I do know enough to talk for an hour
Right.
or two. So anyway,
Well, I think I'm gonna look at this again, and then and then
okay,
I'm gonna see what you did. I'm gonna ask you some questions. I'm gonna do some research because it's
yeah,
something you said earlier. I wanted to tap into, but I ended up forgetting what it was.
okay.
All right.
So I want to address my chat people for a while. I've got
Okay.
0 likes on this video. I don't know why do I have 0 likes on this video?
Because people
Is the
don't like
like
me.
button is the like button broken? No, you're a likable guy. I like you. Anyway, yeah, I don't know where to go next with this. I hope think about the introns a little bit, and the fact that there are so many of them, and like that amounts to about 30% of the genome is in these introns, basically, or 34% or something like that.
Mm-hmm.
And and think about why we like when we say junk DNA, we never think, oh, that does nothing. It has no effect on anything. We're looking at this thing holistically, the whole chromosome, right? The structure of the whole chromosome. Like where are the genes placed? Are some of them next to each other? Are they close to the centromere? All of that stuff. I mean, it isn't like we're trying to ignore this stuff. We're looking at all of it.
No, well, see,
But
honestly, Mike, you way more honest than other people I talk to. You know, you
thank
way
you.
more honest, man.
Well,
And I
thank
think
you.
that your approach is, you know, you accept what it is. You don't run from it. You don't hide from it. There's a lot of guys that hide from some of the stuff that don't really want to address it, want to act like it's not going down because you know the word junk DNA for a lot of years. And I'm telling you, I used to like talk with people a lot of text chat, and they said that junk was just evolutionary relics, and it was just junk. We don't need it anymore, and this and that. You look at it from a whole different light. You look at evolution in a different light than what a lot of other people look at. So you don't shy away from
Yeah.
the tough questions, basically.
Yeah, okay. Well, thank you. I appreciate that.
Right. So
Okay. Oh, okay. So let's sign off here for now and go get some rest.
right.
I
Yeah, I'm
got, I
tired. I gotta take me a quick nap. All right.
Yeah, I gotta take a nap. I gotta take a nap before I eat a bunch of turkey tonight at my neighbor's house. They invited us over. I'm
Right.
having three turkey dinners this year. So
Yeah.
anyway,
Hey, well,
okay, guys.
take care of yourself.
Yeah. Thank you. Thank you all of you out there who were watching us, and thank you, CBrown. Yeah, and you take care of yourself. Have a good day.
All right.
Okay.
Have a good one. Thank you. Bye bye.
Okay. Yep.
Uh,
And we're doing a goodbye.